Hey, everyone. Thanks so much for coming. Yeah, thank you, thank you. We're still bringing people in from outside, so 2 or 3 more minutes. Thank you so much for your patience. The wrangling is continuing. We'll be right out. Thank you.
Welcome to the San Francisco War Memorial and Performing Arts Center. Please take a moment to locate the exit nearest you. Exits are clearly marked with green lighted signs. In the unlikely event of an emergency, please proceed to the nearest exit and out of the building. Thank you.
All right. Hello, everyone. Thank you so much for coming. I'm Arvind Gupta, Managing Director and founder of IndieBio. First of all, thank you all for coming. I really appreciate it. The team appreciates it. We have 11 incredible companies to share with you today, all changing the world in some significant manner. I want to say thank you all for investing in these companies and investing in our future. In the last 5 years, it's hard to believe it's been 5 years, that IndieBio has existed, the companies have raised $450 million, and our portfolio has just crossed the $2 billion mark. Thank you. And so it's been due to an incredible team that continues to push the boundaries, and I'd like to pass the mic over to Maya Lockwood, who's our communications director, to share a little bit more.
So we're excited that SOSV, our parent fund, has raised $277 million recently. That brings us to $700 million in assets under management. Over $100 million of that is expected to be invested into life sciences and deep tech. Our portfolio companies are in the news. Recently, Jungla was acquired by Invitae to advance genetic variance interpretation. We'd also like to congratulate Memphis Meats for raising $161 million to build a future without slaughter of animals. What you see today here is just the beginning. Our founders are emerging voices in biotech. If you're a journalist or a storyteller in the audience, please introduce yourself. Check us out online at Instagram or at Twitter, and come by and visit us. I would like to now introduce June Axup, our scientific director and partner, who will tell us more about Class 9.
So today we are here to celebrate Class 9 and all of their hard work over the last 4 months. But it takes a village to raise fledgling startups. So we especially want to thank all the mentors, the corporate partners, the VCs, and the alumni who have dedicated countless hours in mentoring these companies. This batch, we doubled down on leadership, doing conscious leadership seminars and team building exercises, because we recognize that leadership comes from within. And of course, we thank the greater community who has come to our events to support the companies and also to discuss the most pressing topics in climate change and life-saving cures to diseases. And thank you for all of you to join our community here today. Next, I want to introduce Parikshit Sharma, our principal, to talk about a really exciting announcement.
Hi, everyone. So today, I'm really proud to announce the launch of our second location in New York City in Q2 of this year. With the highest concentration of life science talent and significant NIH funding, the New York ecosystem is ready to take off for commercialization of biotech through early stage venture. And we are ready to go in there and galvanize this forming ecosystem and transform New York into a life science hub on the East Coast, just as we've been doing here in San Francisco. I'd like to take this opportunity to thank our partners, the State of New York and Partnership Fund for New York City, for their support, which will also allow us to launch our pilot therapeutics program, increasing our deal offering up to $2 million per therapeutic company. And above all, today I'd like to invite you guys. I'd like to invite you to engage, to invest in, and to cheer on the coming classes of New York startups with the same excitement, same encouragement that you've always shown us here in San Francisco. Thank you. And with that, I'll hand it over to Alex to share a bit about our global outreach beyond San Francisco and New York.
So one of the things we spend a lot of time doing is actually going out and talking to scientists about starting companies, because it's not the traditional path for many. Besides a lot of industry events, last year we did a lot of roadshows to universities, actually meeting with scientists on the ground, going to their labs and talking to PIs. And we're really excited. 2020 already has quite a few on the schedule. So we'll be going to all the universities you see on the screen. And actually for anyone here who's engaged with a different university who wants us to come, please let us know. And part of that is we are looking for applications. With New York, one of the big things we're very excited about is we're actually going to be doubling the throughput of companies that come through IndieBio every year. So right now we're doing up to 30 companies, and next year we're going to be doing actually up to 60, including New York. So with that, we're really opening up a lot more ideas and a lot more verticals. And a few areas I want to call out that we're excited about and want to see more entrepreneurship in across planetary and human health are, first off, waste to value. So especially in reducing emissions and beginning to do drawdown, looking at greenhouse gas turned into value, and also taking raw materials and waste products before they become emissions and making products out of that. We're looking a lot into regenerative agriculture as a huge impact on both our climate as well as human health and nutrition. So for that, we're thinking everything from the soil to sprays onto plants, genetic modifications, and also transient regulators of gene expression. And lastly, a big area for us upcoming is oceans and water. So that could be desalination or bioremediation, as well as looking at really massive global problems like ocean acidity, polar ice caps, and ecosystem restoration. And in human health, fertility has been a really big area for us. We've been making a lot of connections, talking to a lot of teams. So thinking both of helping conception as well as contraception. RNA as new ways of detecting and monitoring disease, as well as new therapeutic modalities. And lastly, a big area for us is expanding frontiers of neuroscience, and especially focused on mental health, which is a huge crisis and a big underserved disease area that we really want to be helping more entrepreneurs in. So lastly, I'm going to hand it over to Poe Bronson, our strategy director, with one more big announcement.
So small announcements to make. Arvin, come on over here. So Arvin and I wrote a book. Thank you. It's not about IndieBio, but it is began where we were strategizing how to take the core mission of IndieBio and spread it farther and wider. Experimentation as a way of life, the courage to challenge the status quo. And we wanted to get this message to people who aren't just investors and aren't scientists yet. And so much like we would reprogram a mature cell into a stem cell, we went back to the real origin and spirit of IndieBio, to the fun, the joy of just questioning why things are the way they are. Can we do things differently? I'm a questioner. Arvin's a questioner. I think you're all questioners. That's partly why you're here. You feel that call. And so in this book, we ask a few interesting questions, and we run experiments to learn. Arvin and I trade off in the chapters, sending each other out on challenges, and in that way, the book becomes kind of a conversation between us. Our editor, Sean Desmond, published Sagan. He published Feynman, as well as iconic books like Fight Club and Trainspotting. So the praise and the support of the book that he's giving us really means something. I'm excited to say that just this morning, we confirmed that Decoding the World will be followed by 2 more books to become a trilogy. Now, the startups you're about to see here today, the founders, their journey to this moment began as questioners, as asking, can we do it differently? Well, let's find out. Welcome to IndieBio 9 Demo Day.
Hello, everyone. My name is Shane Kilpatrick, founder and CEO of Mumbio. In America, every 2 seconds, someone receives a life-saving blood transfusion. Blood is essential to every healthcare system, a prerequisite for numerous life-saving interventions, including cancer treatment, trauma, and many surgeries. But the nationwide inventory for blood is at best a 12-day supply, regularly dropping below 2 days in times of low donor turnout. Mumbio is on a mission to fix this, not with more volunteer donors, not with any blood donors at all. We're going to fix this by growing blood in a bioreactor. Transfusion medicine is far more complex than people realize. Some patients require chronic transfusions every few weeks, each one lasting up to 8 hours. And it's a secret of the industry that they can develop an immune reaction to the very blood they need to survive. They can't just be given universal O-negative blood. It's a huge pain point for the hospitals and blood centers to find donated blood with the matching antigen profiles required to prevent immune reactions. Our first products will be for these chronic transfusion patients, specifically those suffering from sickle cell anemia and beta thalassemia. Two features make our NeoBlood stand out. First, NeoBlood is a low antigen blood product. It won't cause immune reactions. Second, blood cells last for 120 days. When you receive a transfusion, you have no idea how close those cells are to their expiration date. Our NeoBlood cells are newly formed. They last longer in vivo, reducing how frequently a patient requires a transfusion. This drastically reduces the cost of care. We'll sell NeoBlood for $1,500 a unit. That's a little more than what people pay today, but the cost of care benefits make it far more economical. With a cost of goods under $500, we'll be able to capture value with healthy margins in this $1.2 billion beachhead market. But chronic transfusions are just the beginning for MemBio. As we scale, we'll grow universal O-negative OmniBlood for $100 a unit. That's less than what is spent today to collect and store donor blood. We'll sell a better blood at a competitive price, dominating the $40 billion global market. And once there's an unlimited supply of blood, the real market size is more like $60 billion. Our process starts from hematopoietic stem cells. With a genetic switch, we control when they divide and grow, and we control when they switch over to becoming red blood cells. So we grow them to high density in our bioreactors, then trigger the switch. In the process of becoming red blood cells, they'll enucleate, which is to eject their nucleus and all the genetic material it contains. This ensures none is transferred to the patient. For decades, researchers around the world have been growing blood outside the body. However, none have broken out of the research lab due to high production costs. Now is the right time to leverage this background and commercialize it because MemBio has invented a new, cost-effective way to grow cells in bioreactors. We drew our inspiration from the human body's circulation system. It's the most efficient logistics system ever created. All of the over 37 trillion cells in our body are no more than a fraction of a millimeter away from a capillary and are constantly being delivered the optimal amounts of both gas and liquid nutrients. This is how our body can sustain cell densities simply unachievable ex vivo. Based on this realization, in my background in biomanufacturing and ecology, we designed a new bioreactor based on nature's logistics system. We call it dual fusion. With two systems of hollow, semi-permeable fibers, no cell in our reactor is ever out of range of all the nutrients they need to thrive. But it's far more than that proximity. When our cells are expanding, they prefer a low oxygen environment. When they're maturing, they prefer a high oxygen and high temperature environment. So we made dual fusion a fully sensing system. And at any location in that three-dimensional space, we can optimize the pH, temperature, and nutrient concentrations using precision fluidics. This is data from our international patent application. We can get oxygen to cells 320 times faster than a standard hollow fiber bioreactor. This allows us way more precision in controlling every cell's microenvironment, not only ensuring productivity and product quality, but drastically reducing production costs by unlocking previously unattainable cell densities. That is our unfair advantage. And it's scalable to commercial volumes without any compromises. We've had a regulatory consultant working with us full-time for the past 2 months. She's confident we'll be evaluated as a combination product. This is good news. It means we don't have to file 3 separate applications for our cell line, our device, and our blood product. We submitted our first meeting request with the FDA to confirm this, and we'll have an Interact meeting this fall. We have a long road ahead, but believe now is the right time to de-risk our path to commercialization. During IndieBio, we set 3 milestones to test this, focused on our bioreactor, cell line, and blood production. I'm proud to say that we've hit all 3. Andrew scaled up and re-engineered our bioreactors. Moen expanded our fluid dynamic design from a single hollow fiber in 2D to a full bioreactor in 3D. And my co-founder, Megan, has developed methods for generating specialized cell lines and has started our custom media formulation. She's also performed early optimization of our manufacturing process, which has allowed us to go from producing a speck of blood to over 1.5 milliliters in a matter of months. And we have no plans on slowing down. With our seed funding, our major milestone is to demonstrate cost competitiveness by optimizing our production process. On the regulatory side, we'll perform in vivo studies to demonstrate the safety and efficacy of our NeoBlood. And with the Series A, we'll do scale-up and the IND-enabling studies to become a clinical stage company. I'm excited to announce that our $6 million seed round is being led by a top-tier investor and is already over 50% subscribed. I invite you to join us in revolutionizing transfusion medicine and to fuel life with blood. Thank you.
Hey, everybody. Thank you very much. My name is Doug Manofsky, and I'm co-founder and CEO of Panda Nutrition, and I'm very excited to share with you today how we are greening the livestock industry. Antibiotics were banned in 2017 by the FDA for use as livestock growth promoters. This took a 7-kiloton market for antibiotics and drove it into the ground. This is a huge step forward for human health, but ultimately it presents a challenge for livestock producers. Livestock producers are now desperate for novel, non-antibiotic growth promoters. Central to this issue is the microbiome. For example, the chicken microbiome contains good bacteria and bad bacteria. Antibiotics are overkill, and they completely wipe out the entire microbiome to promote livestock growth. We found a way to preserve the good bugs while we target the bad bugs to promote livestock growth without antibiotics. Our secret is lysozyme. Chickens make lysozyme in their eggs to protect their eggs from bad bacteria. You and I make lysozyme in our tears to prevent eye infections. Lysozyme is the first line of defense an animal has against bad bacteria. Unfortunately, lysozyme is a complex protein. This means it's difficult and expensive to manufacture, and that makes it uneconomical for livestock applications. So we recruited the microbiome to manufacture lysozyme for us. To engineer the microbiome, we start with baker's yeast. Baker's yeast is a safe, common probiotic used in animal feed. In fact, 1/3 of all yeast ends up in animal feed. Using recombinant DNA, we engineer baker's yeast to secrete natural lysozyme directly in the gut of the chicken. We call this panzyme, and it turns the chicken microbiome into a lysozyme factory. While at IndieBio, we validated our approach using 2 chicken studies containing 216 chickens each. And in both studies, we saw a 10% increase in growth over our negative controls. This is a traditional non-engineered yeast as our negative control. We also beat the gold standard antibiotic growth promoter, bacitracin. This means profitable livestock without antibiotics. Antibiotics have been used in livestock production for 70 years, but it is finally over. Using blood serum analysis, we validated that our chickens were in outstanding health. Our animal researcher even said that this was the best group of birds he's ever had. We tested panzyme in chickens because it was the fastest way to prove that it works. But it will also work in cows. And this is very exciting because it offers one important additional benefit: methane reduction. As you can see, by peering into the microbiome of our chickens in our study, panzyme successfully targets ruminococcus. Ruminococcus is key to the methane cycle in cows. If you haven't heard, cow methane is a major contributor to climate change, and beef and dairy producers are facing intense scrutiny. By knocking down ruminococcus, we starve methanogens of essential metabolites. What we have here in Panzyme is an MVP capable of reducing methane emissions while simultaneously delivering the animal growth rates that the industry requires. The animal feed additives business is a huge market, and yeast isn't just a common ingredient, it's the fastest growing ingredient in this market. Here's our plan for the future generation of Panzyme technology to completely eliminate methane emissions. By combining Panzyme with 2 other enzyme-secreting strains of yeast, we will both knock out methanogens and replace their function in the microbiome. We've already met with the FDA, and as long as we are using enzymes, they're supportive of our approach and are generally recognized as safe determination. We are targeting market approval by the end of 2020. We're also developing a specialty strain of Panzyme for the USDA. We are speaking with 3 of the top 10 meat processors in the world and 20+ independent farmers. We are about to close our first LOI, and as we get into field trials in cows, we anticipate many more. Craig Rowski is my co-founder and chief scientific officer. He is a well-known microbiologist and immunologist in the Bay Area, having co-founded the Real Vegan Cheese Project and BioNascent, among other exciting projects. My name is Doug Manofsky, and I'm co-founder and CEO of Panda Nutrition, and I am an engineer and former business consultant turned entrepreneur, and we are both passionate about fighting disease, hunger, and pollution. We have assembled a team of key advisors. Thomas Nagy is the former president of Novozymes North America, a key livestock enzyme manufacturer. Dr. Sandeep Vani is a fermentation expert from Archer Daniels Midland, one of the largest agri-food companies in the world. Together, we cover the entire supply chain. We have concluded our proof of concept research, and we are raising $3.4 million to continue to develop our MVP and get market approval over the next 12 months. We hope that you will join us and fight disease, hunger, and pollution. Thank you. Thank you.
Hi, I'm John Muser, and I'm the CEO and co-founder of Kyber Botanics. We are the plant cell company. And today, I'm going to invite you to think differently about plants. All around us are plant products, but you may not realize the most valuable aren't in the produce section. There's hundreds of products that are high value because they're so difficult to grow. And they make up a $250 billion industry for ingredients in cosmetics, health, household products, fragrances, and flavors. For example, raspberry ketone. It's the essence of raspberry. It's used as a flavor and as an ingredient, but it's present in the raspberry at such tiny quantities that it's worth $20,000 per kilogram. Or aloe. You probably don't realize that the aloe powder used in cheap consumer products is actually very expensive. Processed aloe powder costs manufacturers $400 a kilogram. Or the $60 billion market for natural rubber used in car tires and aircraft tires, but it's destroying our rainforests. So at Kyber Botanics, we asked a simple question. If all we're after is just a tiny part of the plant, why are we still growing the whole plant? What if we could grow just the cells that matter? We can. We can grow high-value products from plant cell cultures the same way bacteria, fungi, and animal cells have been used. But importantly, we can grow high-value products with plant cell cultures that can't be made by these systems. And the key is that plant cell cultures can grow 30 times faster than the same plants in the field. We start the process with the plant. We convert it into stem cells. And once we discover the right combination of growth factors and environmental conditions, they start to become a plant cell culture. But plant cell cultures still need to be improved before they can be scaled commercially to make the final product, which in this case is a non-toxic aloe powder. But we don't do those improvements manually. We use high-throughput robotics to continuously select for just those few cells that grow fast and that make the products we want. Our go-to-market product is aloe. Now, it's the ideal market because it's massive. It's a $1.6 billion market, but it has a problem. Aloe produces a potent toxin. Normally, it has to be processed out. But our aloe vera cells can be grown without the toxin. So it doesn't need to be processed, and that ultimately, it's worth twice the value. This is an aloe farm. It grows slowly. Aloe plants double maybe once a year. But plant cell cultures double on the order of days, not years. So we can replace a whole farm with just a 50-liter volume of reactor. And the aloe market is growing at 10% per year. We'll first intersect the cosmetics and medical market. Then, as we scale up and our costs go down, we'll come into the nutrition, personal care, and eventually the low-cost drink markets. And we're concurrently growing multiple different products, each with a clear path to market. From our fruit stem cells, which are the hottest things in cosmetics, to a more sustainable form of natural rubber, to orange oil, which is used as both a fragrance and industrial solvent, or the highly prized fragrance of vetiver, which is an oil that's used in only the finest colognes. But we started with each of these because we've already identified interest from a customer or an industry partner. So what's important is that we already have revenues. It's because we have a hybrid business model. So we either sell our platform as a service, or we sell the products that we make. We already have 4 cooperative development partners, and they're willing to pay us $1.5 million over the next 2 years, which includes the development of our robotic pipeline. And this revenues that we'll be able to leverage into becoming a product company with many product lines. We already have 3 LOIs for our aloe. We're also working with the product development team of a cosmetics company to help us develop our berry stem cells. Our entire stack is protected, from the cell lines we create to the way we made them, to the way we optimize robotics for plant stem cells, and finally, the genetics of the cell lines we create. Our management team is composed of myself and Robert Jinkerson, CSO and co-founder. We met working together as lab mates in graduate school. Afterwards, I went into industry to learn high-throughput biotechnology techniques. Robert went to the Carnegie Institute of Plant Bio at Stanford, where he adapted yeast robotics to make a 200,000 mutant library in algae. So now we're combining my high-throughput bio background and Robert's robotics and applying it to plants in a new way. We have an esteemed advisory board, including Ben Moll, who's an expert in plant biotechnology, John Feiner, whose specialization is plant cell cultures and breeding, and Mark Fallouz, who's one of the world experts in scale-up of plant cell cultures. And finally, Marquita Landry, who's a UC Berkeley professor who's developed a plant-agnostic way for plant transformation. This year, we're going to be developing our robotics pipeline, and we're going to be scaling up our cultures to the 300-liter scale so that we can provide to our customers sample products and convert those LOIs we have now into JDAs. Our next step is we're raising a $3.75 million seed round. And with that, I'd like to invite you to join us to change how we grow. Thank you.
Hi everyone, my name is Ricky and I'm the CEO and co-founder of Mychroma. Take a look at this bacteria under the microscope. Or is it larva of an insect? Actually, these are Cheetos. And what about this picture? Is this wood, countertop marble, or chewing gum? This is America's #1 veggie bacon. From junk food to healthy food, virtually everything is artificially colored because processed food without dyes look disgusting, not fresh, nor nutritious. And they are everywhere. They are in your candies and soda, but also in your yogurt and pickles. That translates into a $5 billion market, which most of it are toxic petroleum-based dyes. They have been related to many health concerns, ranging from allergies, like the skin rashes I experienced firsthand as a kid, to ADHD, hyperactivity, and even cancer. Yellow number 5 is just an example of the many petroleum-based dyes you and your kids are eating. And this information has been hidden in complex labels. No one, especially the new generations, want to eat these products or even read these labels anymore. The small print is becoming the big print. This is the label of the fastest growing bars brand, recently sold for $600 million. The food industry is suffering because people want clean labels, and they can't have clean labels when adding petroleum-based dyes to their products. Mychroma solved that problem by making food dyes from food that are good for you and the planet, unlocking the possibility for the entire food industry to make their products naturally more appealing and at the same time make higher profits. Let me walk you through how we take nature and we make it better. We selected a wide collection of the best dyes producers in nature, filamentous fungi. By CRISPR engineering them, we created proprietary, non-GMO industrial strains with highly improved yield while also nutritional and functional properties. We grow them in scalable bioreactors with the optimal conditions to make the fungi produce and secrete the color to the media. And after a simple separation and formulation process, we get our bright and beautiful colors. While at IndieBio, many of the top and fastest growing companies have shown interest in Mychroma. We received 5 letters of intent from huge companies like Danone and multiple MTAs from the biggest players in the food, beverages, confectionery, and cosmetic industry. Let me show you the metrics that are important to them. Food is very diverse, from acid to base. Soda is pH 3, while sausages and yogurt are pH 6. And some food even requires to go up to a higher pH and then go down. Mychroma's colorants have the best pH stability when compared to all natural options from insects and plants. While cooking an alternative meat or beef burger, you want it to be bloody red. Plant-based dyes discolors after 5 minutes of cooking. Insect-based dyes are pretty good at pH 6, but they are made by grinding up bugs, so therefore are not vegetarian, kosher, or halal. Mychroma's colorant have the best performance again, and that's why companies are really interested in our products. Our go-to-market strategy is selling our dyes B2B. First, replacing the insect-based dyes, then going for the plant-based ones before targeting the whole dyes industry. While at IndieBio, we increased our yield 74 times. And scaling it up with no more improvement, we could easily achieve our first objective with 80% margin. But that's not enough for us. With our engineer strain, we will be able to achieve a price competitive with the petroleum-based dyes, revolutionizing the entire dyes industry in a cost-effective and sustainable way. We protected this technology working with great IP lawyers, filing provisional patents on our strain and also application, and we are keeping our fermentation process as a trade secret. On the regulatory side, a color petition usually takes 2 years, but we are developing a regulatory strategy to go faster to market. And our great team from the best Latin American universities is highly diverse and experienced in microbial fermentation, molecular biology, food science, and business. And we are honored to be advised by a great team of global experts as well, in every key part of our business and science, allowing us to tackle all the challenges we have ahead. Currently, we are finishing the optimization of our color production platform and testing it with our red strain, to then apply the same technology to produce the rest of the colors of the rainbow. We are raising $2.5 million to take our technology to the next level, finish all the safety tests, and scale up our production. Let's color food with food. Join the color revolution.
Hi, I'm Alex Martinez. I'm the CEO of Lupa Bio. We're going to expand the $50 billion anti-inflammatory drug market by developing a new drug called 3SL for those not served by today's options. 30 years ago, 1 in 400 people suffered from an autoimmune inflammatory disease. Today, it's 1 in 12, and increasing year over year. My co-founder and I witnessed this problem emerge across society while working together in the pharmaceutical industry. Unfortunately, all current treatments have serious side effects and even life-threatening risks. This is a black box warning. It's the FDA's strongest danger sign. Almost all anti-inflammatory drugs have one. It's to warn patients that the drug they're about to take requires a serious trade-off to their health. As a result, these drugs are only used chronically in the most severe cases. For the majority of patients, however, these side effects offset the benefits, leaving them suffering but unserved. We think we found a solution, and the story begins 80 years ago, during a time when there were no effective anti-inflammatory drugs, when scientists began searching for the reason why women with inflammatory disease would often go into remission during pregnancy. They never did find what they were looking for, but almost a century later, a clue was found. Sugars in human milk, called human milk oligosaccharides, HMOs for short, were found circulating in the blood of pregnant women. This was surprising because previously these HMOs were really only thought to serve as prebiotics for the infant microbiome and anti-adhesives for pathogens. So this observation begged the question, what direct effects might these have in the body? Researchers at UC San Diego evaluated all 200 pooled HMOs and saw direct anti-inflammatory effects, indicating that at least some of these sugars work in both moms and babies like drugs. They then went on to identify 3SL as the single HMO primarily responsible for these effects, and perhaps solving this age-old mystery. Incredibly, 3SL is not just safe, it's 5 times less toxic than baking soda in animals. And it's also safe in people. We have safety and tolerability data equivalent to a full phase 1 study. So how can 3SL be anti-inflammatory, yet so much safer than other drugs? Well, it doesn't work by suppressing the immune system like those other approaches. Think of it as Mother Nature's switch to resolve inflammation. You see, when a macrophage is activated by signals from tissue damage, it goes into attack mode, which only leads to further destruction. When 3SL is introduced, it works through the EP4 receptor to resolve inflammation, telling that macrophage to go from attack to repair mode, which should lead to relief from disease. So now with the understanding of that mechanism, the next step was to see if it worked in animal models of inflammatory disease. This is a mouse model of rheumatoid arthritis. Note the pronounced swelling in this untreated paw. Now with 3SL treatment, swelling is reduced. The paw is essentially normalized. This tells us that 3SL holds promise to address symptoms of inflammatory disease. But beyond swelling, when we look at the tissue inside of the paw and joint, we see true disease-modifying activity. So looking at the blue column, generally, when you see something white, it's either healthy cartilage, connective tissue, or bone. So starting at the top, we see that 3SL repairs cartilage. Next, it clears inflammation from the joint. And finally, it eliminates pannus. This is a mass of inflammatory cells. You can see it in the purple on the left in the untreated mouse. This pannus causes bone erosion. If you're someone with this disease, you don't just want the swelling to go down. You want your body to repair itself. And that's exactly what we're seeing in our data, and it's why we're so excited about 3SL as a drug. Because of the wealth of safety data supporting 3SL, we were able to use the FDA's 505(b)(2) pathway to go from an academic license to submitting our full pre-IND briefing package in a little over a year. In 2 weeks, we're going to have FDA feedback on our clinical path with the goal to initiate human studies as part of our phase 1b/2a program within a year. To go fast, we identified a rare pediatric disease with a true unmet need, and this is for kids with a subtype of juvenile idiopathic arthritis, in which there are no approved branded drugs because of their toxicities. If all goes well, we'll know if we have an effective drug for these kids within about 3 years, and from there we can rapidly expand to much larger opportunities like adult rheumatoid arthritis. A truly safe and effective anti-inflammatory drug is a pipeline within a product, so this is just a beginning for us. Our IP comes in the form of an exclusive therapeutic license from UC San Diego to a broad worldwide patent application. And of course, we're filing our own internal IP to further protect and create opportunities for the business. My co-founder and I worked at Ionis Pharmaceuticals on the business, patent, development, and regulatory side of over 40 clinical stage programs, including 4 now approved drugs. This experience, combined with our training as attorneys, enables us to go from strategy to execution extremely rapidly. And in short order, we have forged the components necessary to become a clinical stage company. We have physician clinical advisors, world-class key opinion leaders for our initial indications, former FDA regulatory experts, leading clinical research organizations, and even the supply chain necessary for our initial human studies. I was the first 6-figure check in, followed by IndieBio, and we are welcoming new investors to see us through our FDA interaction and enable us to continue executing on our IND filing and clinical study startup activities. Following formal FDA feedback, we will set our initial equity raise to fully fund our first in-human study. So, speaking as a founder, an angel investor, and a patient, I ask that if you share our vision of developing a drug that doesn't require those suffering from inflammatory disease to accept side effects for relief, let's talk. Thank you.
Hi, I'm Verge Khan, CEO and co-founder of Primitives Biodesign. Primitives is a materials company that develops films for next-generation packaging. We live in a hyper-convenient world. With a click of a button, I can have my dinner for the night, groceries for the week, delivered to my door. Vegan shoes from Finland or locally produced cannabis. This growing modern lifestyle where things arrive in an instant also has a long-term cost, plastic packaging. And they're here to stay with every click, swipe, and purchase we make. Environmental issues caused by plastics and fluctuating costs for petroleum has spurred widespread demand in the bioplastics industry. Everywhere there's a solution, it's rapidly being adopted, growing over 20% a year. Beyond that, the margins for this market are much higher than that of petroleum-based plastics. One area that remains underserved by biodegradable solutions in the plastics industry are flexible films, which is a $250 billion market. These films contain over 5 layers of plastic, and they cannot be recycled. In recognizing this, we've utilized nature's library of renewable materials to create high-performance, compostable films that are safe and even edible. We know this is a competitive market, and now I want to show you what we can do that no other company can. Because we're leveraging biology, we can incorporate properties like sensing and response to its environment. We're inspired by chameleons that change color, plants that emit odor in response to insect attack, pine cones that change their shape to release seeds of the most optimal humidity. At MIT, my co-founder Noah Mackover and I engineered a materials toolkit, creating films that can sense the environment and display information. We've applied this to create sustainable products that can solve our customers' problems across industries. For example, supplement packaging that communicates when it's been tampered with. Cannabis packaging that you can bury in your garden after its use. Insulin labels that respond to extreme temperatures to let you know it's no longer safe. Milk that tells you it's gone bad without having to taste or smell it. Hundreds of consumer packaged goods companies have made public commitments to reduce their plastic usage. Our first customers have given us their spec sheets and told us what mattered to them. And we've engineered films that meets their needs and even outcompete our competitors. Our customers want to improve the freshness of their products. Our films provide an oxygen barrier that's over 4 times better than petroleum-based films like LDPE and bioplastic films like PLA. Beyond that, they can protect and block 98% of harmful UVB rays, which is comparable to wearing sunscreen with SPF 30. Plastic ban regulations are becoming more and more explicit about how fast packaging needs to degrade. Our films degrade faster. They're compostable within 10, 20, 30 days, you choose. Fast degradation of 70% or slow degradation of 20%, mass loss over 3 weeks. Our customers also want to be able to customize their packaging to differentiate from their competitors. Our films are colorful and span the entire visible spectrum. They're optically transparent and can also be printed onto. Our go-to-market strategy will begin with a 100% compostable, oxygen-proof film sold to the premium cannabis packaging market. This market is high margins and also has a growing demand in new packaging innovations due to regulatory requirements. Next, we'll offer functionalities that improve product freshness and safety. Later, we'll provide sensing for environmental monitoring. We know cost is a problem, and we have a plan to reduce it. When we scale to pilot, through automation, we'll be able to reduce our cost by over 2,000%. We will sell 1,000 square foot rolls for under $400, which is comparable to premium films in the cannabis packaging market. Next, when we scale to production, we'll optimize our film thickness. increase our yield, and expand our R&D efforts to incorporate agricultural waste-based feedstocks, which farmers currently pay to dispose of, and take advantage of methane reduction subsidies. Here, our material scientist, Becky Wilson, is performing a Meyer rod test. This is an industry standard for assessing the compatibility of films with extrusion-based processes in manufacturing today. This is an essential step to proving the viability for us to scale with minimal barriers. Our IP portfolio consists of proprietary compositions for engineering films with functional and stimuli-responsive properties for applications in the health and wellness space. After IndieBio, we'll conduct our pilot tests, attain certifications, and launch our first product by the end of this year. Next, we'll target the supplement packaging market and begin moving into production scale. And we have the all-star technical team to do it. Becky has a PhD from USC who has a deep knowledge in material science and chemical engineering. She's also previously co-founded a biotech startup. Noah Mackover has a background in synthetic biology, design, and advocacy from Brown University and MIT. I previously designed flagship products at Samsung and engineered robotic interfaces at the NASA Jet Propulsion Lab. Our advisor, Lee Bowman, has over 15 years of experience in roll-to-roll functional film manufacturing from Eastman Chemical Company. We're raising a $500K seed, and we're hoping that you'll join us in building a world where the materials around us are intelligently engineered and don't overstay their welcome. Thank you.
Hi, I'm Mickey Pentecost, co-founder and CEO of Diadem. We make exosome therapeutics. Cells that are touching are in constant communication. But for cells to communicate at a distance, they secrete extracellular vesicles called exosomes. Originally thought to be cellular garbage, exosomes are now a hot new area for therapeutics. Our competitors are focused on what they can put inside of exosomes to deliver inside of cells. We're focused on engineering the outside of exosomes to control cells through their surface receptors. To do this, we engineer the genetic code of immortalized human cells so that the exosomes come out with therapeutically valuable signaling molecules on them. We know their exact size and concentration, and we even know how many proteins we're putting on the surface, ensuring consistent manufacture and dosing. By engineering the surface of exosomes with signaling agonists, we're creating a new therapeutic modality that will eclipse cell therapies and biologics. I say this confidently because of a simple concept we discovered at IndieBio. When we put a signaling protein on the surface of an exosome, it is 10,000 times more potent than the unattached protein. Let's look at this in depth. PD-1 is a co-inhibitory receptor that controls the activity of immune cells. On the right are two purified proteins that bind this receptor. You can see they cause some signaling, but not very much. In contrast, when we put these same molecules on the surface of exosomes, they become very powerful and robust signalers. And they do so at a drastically lower concentration and dose. This is the difference between a research tool and a therapeutically viable platform. Now let me explain why being able to do this is so important. On the left is receptor superclustering, where two cells interact. Since the development of human monoclonal antibodies, pharma has been trying to engineer ones that cause the same stimulatory agonistic signaling. They've gotten really good at blocking signaling, but they can't get this agonistic effect. They can't get the receptor superclustering. What we've engineered here, the ability to do just this, is a game changer for biotechnology. We did all of this at IndieBio, and with the little time we had left, we performed a proof of concept study in animals. We hadn't optimized our dose at all, we were just looking for a signal. Let me walk you through our data. We immunized rats with a retinal peptide, and it results in severe autoimmune inflammation of the eyes. Disease starts at about day 6 and peaks around day 12. Injection into the eyes of saline vehicle, or our unmodified exosomes, have identical disease progression. In contrast, when we injected our engineered exosomes, it significantly suppressed the progression of disease. As expected, however, there was a half-life, and the effect wore off. But a second injection also appeared to have some effect on the rate of resolution. And we saw nearly identical effects when we injected our engineered exosomes intravenously. In addition to our PD-1 agonist exosomes and optimizing other signaling agonists, we're now making multi-specific exosomes with combinations of therapeutically viable signaling molecules on their surface. The potency and versatility allows us to target a multitude of diseases, including cancers, infectious, and neurodegenerative diseases, just to name a few. There are too many for us to target alone, so we're developing assets that we can partner out, and our internal pipeline will focus on inflammatory diseases with unmet needs. So how do we make them and deliver them? We've been hard at work at IndieBio, harvesting, concentrating, purifying exosomes at lab-feasible scales. But we've also worked with a leading bioprocess lab to create a cGMP-compliant, scalable bioprocess for our upstream manufacturing of exosomes from our cell lines. This upstream process uses no animal-derived ancillary materials, ensuring that we can easily isolate human exosomes, and also that we can, for regulatory considerations. In order to produce our exosomes consistently and affordably, we took advantage of the fact that exosomes are the size of viruses. We realized we could apply vaccine manufacturing technologies to their downstream purification. And at IndieBio, we developed our own workflow to separate exosomes from other biomolecules produced by the cells. Importantly, exosomes are more affordable, stable, and dosable than cell therapies, which are also a logistical mess. And because exosomes are remarkably robust entities, we're working on various delivery formats, including inhalable. This opens up opportunities to target rare and common diseases, some with huge markets. We are so confident in our platform and our therapeutic modality that we welcome the biotech industry to come partner with us. We're particularly interested in treating chronic obstructive pulmonary disease. COPD is a growing global problem and the third leading cause of death by disease. It results in nearly $50 billion of direct medical costs per year in the US. And despite having a $14 billion pharma market, there are currently no disease-modifying therapies. We've assembled amazing scientific, clinical, and business advisors to help us reach our goals. Diadem is co-founded by Dr. Wojtek Barkowski, a brilliant biologist and technologist. He and I have been great friends and collaborators for a decade since we were postdocs together in the same lab. I went on to work in biotech, and Wojtek gained expertise in intellectual property as a technical consultant for a leading IP firm. And we've assembled counsel with expertise in building biotech companies and even in prosecuting exosome technologies. And we aim to occupy a large position in this rapidly advancing field. We've mapped our milestones and our use of funds to create tremendous value over the next 2 years.
We are now opening up a $5 million seed round to create the therapeutic modality of the future. Please come talk to us. Thank you.
Good afternoon, everyone. I am Ravi Adgulwar, co-founder and CEO of Aviza Maiko. We have developed a technology to produce melanin from a proprietary strain of fungus. Melanin is this amazing pigment which gives our skin and hair its color. Its evolution's answer to radiation, and it protects our body from the harmful solar radiation and makes life on this planet viable. Melanin also is a multifunctional supermaterial with amazing industrial properties. It is an excellent blocker of UV radiation and hence can be used to make effective sunscreens. It blocks ionizing gamma radiation and X-rays, and hence it can be used to make shielding against radiation in satellites, aerospace industry, and nuclear applications. It can be also used to protect the patients and the medical staff involved in radiation therapy. It has got amazing electrical properties. It is a rare organic semiconductor and can be used for implantable electronics and biosensors. Carnegie Mellon University has developed a melanin-based cathode to make low-cost sodium-ion and magnesium-ion batteries rechargeable. Like solar panel, it can be used to generate electricity from sunlight. Cooper University has demonstrated this with our melanin. So if melanin can do all this, why haven't we heard about melanin? Why don't we have melanin-based sunscreen and melanin-based power plant? The reason is melanin is very costly. It is currently sold at $400 per gram. That is 8 times costlier than gold. Researchers have been trying to make melanin with chemical methods and by recombinant methods for the last 20 years. They are only able to make in milligrams and micrograms. We can make it in kilograms. Over the last 10 years, we have optimized our process, and now we can make melanin at 20 cents per gram. That is 2,000 times lower. And this makes many of the industrial applications, which were earlier not commercially viable, now commercially viable. At IndieBio, we have been working to scale up the process, and now we have validated our process at 50-liter scale bioreactor. Very soon, we will be taking trials at 500-liter scale bioreactors. The next step after the scale-up is application development with our industrial customers. We are working to formulate a sunscreen with our melanin. As you could see from the preliminary results, melanin has a potential to replace the harmful chemicals like avobenzone, nanozinc oxide, OMC, and other harmful chemicals from the sunscreen. These chemicals are known to damage the coral reefs and the marine life they sustain. If these chemicals are toxic to the coral reefs, can these chemicals be toxic to our bodies as well? There is a widespread concern regarding the safety and the toxicity of these ingredients. US FDA has ordered a safety review of 8 major sunscreen ingredients. This safety review is currently on hold under lobbying pressure. We believe our melanin can be the solution to the current problems being faced by cosmetics and the sunscreen industry. Initial ex vivo tests show no indications of toxicity. And this is not surprising given the fact that melanin is part of our skin and internal body organs. UV filter ingredient itself is a $700 million market size, with dyes and radiation protection markets representing even bigger opportunities. Melanin blocks ionizing gamma radiation and X-rays. These are the results of a small experiment where we expose melanin solution over X-ray film. As you could see, we could block X-ray with a very modest concentration of melanin, and this makes it useful for radiation shielding in nuclear applications, aerospace applications, and medical applications. And this data is pretty amazing. We injected melanin in the mice and exposed them to 7 grayscale radiation. That is 3.5 times the radiation a cancer patient undergoing radiation therapy would be exposed to. After 30 days, all the mice not injected with melanin died, whereas the mice injected with melanin before the exposure, all of them survived. And even the mice injected with melanin after the exposure, 70% survived. So melanin has a potential to be developed as a drug and a prophylactic against radiation toxicity in case of accidental nuclear exposure. We are a B2B company. We are currently focused on selling melanin for UV protection and radiation protection application. In future, we plan to develop electrical applications, but that would be through JVs, partnerships, and spin-out. Long horizon applications like semiconductors and drug would be first developed through research partnerships. We have sampled our melanin to many potential customers, and we are getting excellent response. We have signed material evaluation agreements with the largest aerospace and defense contractor in US, a large multi-billion-dollar membrane and textile fabric making company, and the largest cosmetic manufacturer headquartered out of Europe, and many more companies. We have received orders for more than 1 kg of melanin, which we sold at $30,000 per kg. I am Ravi. My prior experience has been in business development and general management with Tata Chemicals Limited, India. My co-founder, Raghu, is an internationally known marine microbiologist and a mycologist. He has discovered several new species of fungus, and a genus of fungus is named after him. We know each other for the last 5 years and have been working together for the last 3 years. We have Dr. Howard Maibach as our advisor, who is an expert in toxicity and sunscreen. He is advising us on the regulatory aspect. We have Mark Goldstein, the founder of UCSF Health Hub, as our advisor, and he is advising us on the business strategy and fundraising. He is also an investor in our company. They both are here in person to talk with the investors. We are raising $1 million to take this to the commercialization stage. I invite you to join me to unlock the amazing potential of melanin. Thank you. Thank you.
Hi, everyone. My name is Austin Quach, co-founder of Dalton Bioanalytics. We're digitizing blood for the future of human health. These are the biomarkers that make up our annual blood work. There are small molecules like glucose, electrolytes like calcium, proteins like hemoglobin, and lipids like cholesterol. For the past 40 years, these same 32 biomarkers have been used in our routine checkups. Even with all the tests that doctors can order, they're only looking at about 1,000 biomarkers, leaving more than 4,000 blood biochemicals unexamined. The complete view of all of these molecules, known as multiomics, has been shown to dramatically improve diagnostic performance across a wide range of diseases. Researchers, liquid biopsy companies, and innovative healthcare systems alike are sprinting towards using multiomics for early detection and ultimately better patient outcomes. Unfortunately, it's still a huge pain to run this analysis. You have to split your samples, send them to several specialized laboratories, have a bioinformatics expert integrate the data at the risk of distorting the results. In total, this can cost thousands of dollars per sample. Dalton is a one-stop shop. We measure proteins, lipids, electrolytes, metabolites, and other small molecules for just $500 per sample, dramatically simplifying this entire process. We can do this because we've invented a new way of using tried and true mass spectrometry to measure all types of biochemicals in a single run. We take the macromolecules and we clip them up into smaller pieces. We take the misbehaving molecules and we pair them with chemical chaperones. We digitize this entire complex mixture using high-resolution LCMS, and we reassemble the sample in the computer using bioinformatic algorithms. We've automated this entire process from sample preparation to data analysis and acquisition. The only thing we do manually is move the sample plate from machine to machine. This is a spectrogram showing our analysis of human plasma. Each of the colored specks here represents a unique molecular feature that we find in blood. For example, in the bottom left-hand corner, zooming in more than 1,000x to this specific mass and time, we see the signal for glucose. Over here on the right, at this specific mass and time, is the signal for one of the forms of triglycerides. The center cloud is composed of fragments from over 700 proteins. The dense cloud to the right is composed of hundreds of lipids, and sprinkled along the bottom and left are electrolytes and small molecules. In total, after optimization, we expect to be able to measure more than 2,000 blood biochemicals in a single run. Our method has high reproducibility, with global correlations between repeat measurements greater than 95%. At our time in IndieBio, we've gone on to demonstrate an important proof of concept. Using machine learning, our data can be used to learn new biomarker signatures, getting smarter with every run. Traditional diagnoses are based on just a few biomarkers. Here, highlighted by these boxes in yellow, are all of the analytes we found are associated with aging, cardiovascular disease, inflammation, and dementia. Remember, these aren't separate assays. We can run all of these and more using just one single blood analysis. And every new biomarker we identify holds enormous research potential into its hidden role in disease. This is the value we provide to our customers in the rapidly growing multiomics market. Since our time at IndieBio, our machine has been running nearly 24/7. We have 2 ongoing clinical pilot studies and 2 letters of intent reserving future instrument time. But it's not just the LOIs. We're helping to answer questions such as, can we diagnose kidney transplant rejection before it happens? Congenital heart disease before birth? For those of you with allergies, can we replace steroids with a natural, non-toxic compound? Can we diagnose Lyme disease accurately? We're not just another diagnostics company developing a one-off test. We're partnering with researchers, empowering them to replace existing tests with superior biomarker signatures, and more importantly, to discover brand new tests to address the numerous unmet diagnostic needs that are struggling to be fulfilled. Drawing upon data that we gather from our multi-omic research platform, we'll continue to develop new clinical tests that will expand the existing clinical lab market. We see ourselves much like next-generation sequencing, a complete scan for numerous diseases and conditions. However, unlike NGS, we'll be able to create a low-cost liquid biopsy for rare and common diseases. Long-term, this means that we can readily integrate into existing healthcare practices as a semi-annual blood test. I co-invented this method during my PhD at UCLA, where we demonstrated for the first time in history that we could measure all types of biochemicals using a single assay. Seung-Jun Yeo, our CEO, brings his leadership and experience from his previous venture, developing AI software for prenatal detection of congenital heart disease. Our advisors are composed of world-class experts in analytical chemistry, molecular diagnostics, and medicine. Dr. Kim Fall is co-inventor of the method. He offered us invaluable resources and guidance during the development of the assay. Richard and Yalda continue to shape how we will develop new clinical tests that meet the rigorous standards expected by the medical community. We have full protection of our technology stack with exclusive licensing of the analytical method from UCLA and development of the bioinformatic algorithms in-house at Dalton. We're raising our seed round to build our lab, launch our multi-omic research service, and then we'll get certified as a CLIA lab to run clinical tests for medical providers. We're raising $3.2 million to build our lab. Please come find us to learn how the future of medicine is fueled by digitized blood. Thank you.
Hi, my name is Gabriel Paulino. I'm the CEO of Biolumine, a nutritional technology company for metabolic health and weight management. So about a year ago, I was a senior scientist for Bulletproof, developing their dietary supplement portfolio, when my co-founder came to me with something quite interesting. That's when I decided to leave Bulletproof and start Biolumine. So we all know this. From being a hunter-gatherers to eating our lunch behind our desk, we've evolved technically pretty quickly. However, our body and digestive systems have not, which has led us with an impaired balance, with an impaired health, and making us gain weight. There's one thing that nutritionists agree upon, is that dietary fiber is the #1 nutrient deficiency, and that the balance between dietary fiber and sugar has been completely reversed from hunter-gatherers to modern days. We spent $88 billion just in the last year on trying to find a cure for obesity. Well, we believe we might have found a solution. Biolumine has engineered 100% natural structure functional fiber that can simply turn the food you want into what your body needs. And we do that all naturally. Now let me tell you how it works. Biolumine fiber is odorless and flavorless, and is taken with each meal. Biolumine fiber is a three-dimensional structure of cellulose dietary fiber that has been coated with a blend of smart hydrogel and compressed into beadlets inside of a tablet. So when that tablet dissolves in the stomach, it will expand 20 times volumetrically, and in the process, absorb sugar and starches and contain them with this blend of smart hydrogel. As it expands, it will create a sense of fullness. And as it moves safely through the digestive system, it will control the release of nutrients, protecting the liver from excess sugar, and also protect starches from being digested by digestive enzymes. It will allow the gut microbiome, though, to use the starches as prebiotics, converting them into short-chain fatty acid that will help the digestive system and repair it from inside out. The fiber will then leave the body naturally, just like any other fiber. Meet Chyme, the simulator of human intestinal microbial ecosystem. This is a scientifically validated model of the entire human digestive system, which has been used by the food and pharma industry to test the efficacy of their ingredient as well as their drug, and also test the impact on their gut microbiome. So we tested our technology in this model, and we showed that we can reduce by 1/3 the caloric intake coming from starches and convert those into prebiotic, literally turning the food you want into what your body needs. In terms of expected consumer benefits, we'll expect from the early adopters that have tried a prototype, as well from the results of our preclinical study, an increased sense of satiety and lasting energy, an increase in lipid oxidation or fat burning, and an increase also of a healthy modulation of the inflammatory response due to the production of butyrate, one of the key short-chain fatty acids for gut physiology as well for overall health. All that with no GI discomfort, which is the one main side effect of prebiotic supplementation. This will improve the metabolic health profile of individuals, which in the long run will induce weight control and weight loss. Since we've developed this technology by design with food ingredients commonly used in the food supply, we'll be launching this as a direct-to-consumer brand for $49.99 per month as a monthly subscription service. And we will be driving traffic from social, digital advertising, and blogs to our website for pre-registration so people can know about our technology. And we're happy to announce that our pre-launch website is live, so take your phone out, go to goodforme.com, and pre-register to know about the launch dates this spring. We'll be launching first as a consumer product, and then with an established consumer channel, we'll be able to extend to other product categories. Finally, we will be able to license out our powder form to other food and ingredient companies as a B2B business. As a matter of fact, since we joined IndieBio, a lot of food and ingredient companies have been very interested in integrating this technology in their food product. At IndieBio, we're able to optimize our formulation and go from a lab prototype all the way to a pilot scale that we'll be able to sustain our small volumes for a product launch here in the US. We have a strong IP portfolio with 6 patent issued, 7 more pending in 5 different countries, all around our structure of our technology, as well as its application for reduction of caloric intake, weight loss, and weight management. We also just filed a provisional for the therapeutic application of this technology for diet-related chronic diseases such as type 2 diabetes, as well as the management of type 1 diabetes. We have a very experienced team with many years of experience from the nutraceutical, material science, medical, as well as marketing space. My co-founder, Paolo Costa, polymer engineer, inventor of this incredible technology. Wayne Jashin with 10 years of manufacturing experience. Stephen Davis with 30 years of experience scaling up processes for performance polymers in the manufacturing for medical device. Myself, Gabriel Polino, PhD in neurophysiology of nutrition from UC Davis, studying how the brain and the guts communicate to detect nutrients, and spent the last 10 years in the nutraceutical industry launching dietary supplements with my most recent experience for Bulletproof. Finally, Jason Kelly, bringing 25 years of marketing executive experience launching D2C and B2B brand. We have also a great team of advisors from business and medical sides. To be noted, Robert Lustig, considered one of the top 10 obesity experts in the world, and also a New York Times bestseller author book with his book, Fat Chance. So we're raising a seed round of $3.2 million to finish our product development, go to the US market, organize our first marketing claim study, as well as hire our key team members to take this successfully to market. If you'd like to know more, please contact me at getbiome.tech, or you can scan this QR code. Thank you.
Good afternoon. My name is Luke, and I'm the CEO of Agra Sea. Agra Sea is the ocean agriculture company. Quite simply, we grow rice in the oceans. Today, and for every day this year, 3.5 billion people will rely on rice for their staple food. That's almost half the world's population, with the demand set to double by 2050. And yet, this $311 billion industry is already struggling to produce enough. With the continued climate change we're experiencing, this is what Vietnam will look like by 2030. The Mekong Delta is already being flooded by the rising sea levels, and by 2050, the entire southern agricultural region will be underwater. fresh water is a finite resource. 70% today go towards our agriculture, and rice uses more than any other crop. This has led to so many other countries being in a similar situation. So we decided to adapt, to create oceanic, salt-tolerant crops. We did this by identifying a handful of genes that allow organisms such as mangroves and seagrasses to activate their salt tolerance and thrive in the oceans. Rice has these same genes. So we transformed rice stem cells and accelerated the evolution of those genes using our gene gun, shown here. We can then regenerate those into full plants that have gained the ability to grow in the ocean. Our rice is not GMO, since we do not incorporate any genes from any other species. Our genetic design is based off 4 key pillars that have each been individually proven over the past 70 years through in-depth academic research. Each of these pillars manages the concentration of our salts between the plant and its environment. Our genes, localized within the roots, shuttle the ions through the plant tissues to create a buffer against further salt entry and prevent and pump out any excess salt. During IndieBio, we have transformed over 2,000 plants and are now selecting for oceanic salt tolerance. Over the next 2 months, we will steadily increase the salt concentration, select for optimal varieties, and perform genetic analyses to show stability across generations. Our farm design is being crafted by a world-class architectural firm that already has extensive experience working with floating ocean cities in the South China Sea. This is just concept art, but it shows a few key features, including our catamaran harvester that clips and collects our rice heads, the octagon fields that can unspool or aggregate to mitigate wave stress, or the modularity that allows us to scale to demand rapidly and with ease. These octagon fields can be launched from shore and remotely controlled to be situated anywhere between 1 and 200 kilometers from the coastline. Here, they can take advantage of the abundance of nutrients carried around the world by the deep sea, Arctic, and Antarctic currents. As these currents approach the continents, they rise to the surface. We use technology to enhance this upwelling and create the ideal locations for our farms. My incredible co-founder didn't think this was enough, and so he's developing a bacterial recruitment technology that acts as a methane-negative natural fertilizer. Our unique vision for the future of agriculture has already gained international recognition from some of the greatest governments and largest organizations around the world. This has led to multiple offers for pilot plot locations, including Vietnam, Bangladesh, and even the Bahamas. Farmers today have to pay leases on their land over many years. With Agra Sea's ocean agriculture, you pay a one-time manufacturing fee, you don't use any fresh water, and no fertilizer. This completely changes the economics of farming, making our ocean farms more profitable than farming on land, be that in greenhouses, on farmland itself, or in hydroponics. Because our rice is methane negative, the carbon credits we generate from growing our rice offsets the capex of deploying these farms. Our first act will be to license this technology out to a global agricultural firm that can distribute our plants around the world through existing agricultural channels. We will use this revenue to expand our crop portfolio, honor food security contract from nation states, and deploy our own ocean farms that can allow us to supply multinational food companies. And I'm very happy to announce that we're already in discussions with 2 of the big 4 agricultural companies, 7 nation states, and 3 multinational food companies. Our team is based off a friendship of over 6 years, with myself as CEO and my co-founder Rory as CSO. We met at Durham University in the UK, where we both gained master's degrees in plant genetics and tissue regeneration, respectively. Jude joins us as an investor, having previously been an early investor in Ginkgo Bioworks, Benchling, and Gusto, while Adam brings his expertise of the agronomy and agricultural sector as the Senior Vice President of Revenue at Granular. We've filed our provisional patent with WSGR that has been strategically crafted to protect our genetic design as we expand our crops into the ones most critical to our society today. We're also very excited to be working with key policymakers, both locally and internationally, that can help us create policy that will govern the deployment of our farms along coastlines around the world. Agra Sea started as a dream to feed 10 billion people and eradicate world hunger. During our time at IndieBio, we made the first steps to achieving that dream. By the end of this year, we will have our first pilot plots deployed, and by the end of 2022, we predict that we will produce enough food to feed 1 million people. Our seed round is open at 2.4 million, which we'll be using to expand our crop portfolio, deploy our own ocean pilot plots, and expand the Agra Sea family. That is the core of who we are. Agra Sea is the sustainable future of agriculture. Now let's take the 3.5 billion we can already feed and raise it to 10. Thank you.
All right. So that was class 9. Let's give a round of applause for everyone. As always, an amazing group of individuals and teams trying to solve the world's biggest problems. I'd like to call up Sean O'Sullivan, the man behind SOSV and really the visionary that's brought all these accelerators around the world to solve really big problems. And without his vision, IndieBio wouldn't be here today, very simply. Very few people could put this amount of capital to work and in something that no one really thought was possible. And it's truly amazing to me that people like him exist. So I just want to say thank you for this.
Thanks. Yeah. Every time I come to one of these demo days, and of course I'm here a lot to meet with the teams, it's just amazing to see how much and how much better the future could be than what our experience is. I know, like, even now, you know, we're in tumultuous times. We're living in really difficult, you know, sort of political atrophy and some questions about leadership, and so people look at all those things, and in many cases it's depressing, and it leaves you without hope. And when you look at the arc of time, it does favor the progression of the planet through science, through technology, and the application of really amazing things. Even if some of our leadership is denying climate change, or some of our leadership is working against that, we are actually seeing that the future you can come to IndieBio and see that the future can be bright. You can turn off the television, just come here and help these companies help create the world, help create a better and brighter world. So, if there is anything that you guys saw today, and I'm sure, like, what did we see? We saw a new way of making blood. We saw new therapeutics that are going to actually impact dozens of inflammatory and autoimmune diseases. We saw, new solutions for climate change and planetary and human health. The future was on the stage today, and it's going to be out there in the entrepreneurs that are out there on the tables. I'd love for you to engage with our entrepreneurs. I'd love for you to help see if you can play a part in backing or in making this brighter future more possible. So thank you very much for attending today, and handing it back over to Arvind.
Thank you, Sean. Awesome. So one little last bit of housekeeping. I'd like to ask you all a favor. So the investors have an orange lanyard. The companies outside are here to really raise funding and to meet investors. So for the next half an hour or so, if you're not an investor and you see a company engaging with an investor, please wait a little bit and give them some room to make the introductions and get their meeting set up and all of that kind of stuff. It's rare to have this many people interested in this space at one time, so let's maximize that. We'll be around until 7 o'clock outside, so please, there's plenty of time for everyone to meet. Thank you again so much for coming. I look forward to seeing you next time for Batch 10. Bye, guys.